Alright, and so now we're going to talk about apoptosis. Now, apoptosis is also a mechanism for cellular death, but it differs from necrosis in mainly two things. Now, necrosis is always pathological. However, apoptosis mostly is physiological. And necrosis is always followed by acute inflammation. However, apoptosis is never followed by acute inflammation. So those are the main two differences between apoptosis and necrosis. Now, apoptosis is a Greek term that means the falling off of leaves from trees. So it is considered as a cellular suicide. And the funny thing is that this cellular suicide is energy dependent. So this program of apoptosis and the process of apoptosis is actually ATP dependent. Now, before I go into the details of apoptosis, I want you to remember a couple of things. Now, let us suppose that this is a mitochondrion, and this is the inner mitochondrial membrane. Now remember, we have a molecule, we have something within that inner mitochondrial membrane. It is called cytochrome C. Now cytochrome C, of course, you recognize it because it plays an important role in the electron transport chain. However, the thing is, any disintegrity within that membrane, within that mitochondrial membrane, will cause a leakage of the cytochrome C outside of the mitochondrion and into the cytosol. And this acts as an initiator for apoptosis because it initiates some enzymes that are called caspases. Now, caspases are enzymes that when they're activated, they will activate two families of enzymes that will begin the process of apoptosis. They will activate proteases, and basically proteases' job is to now you remember that the cell has a cytoskeleton that keeps the cell's shape and size intact, and so the proteases will actually digest that cytoskeleton. And it also activates endonucleases. And endonucleases, they will basically digest DNA within the nucleus. Now, actually, if you take a picture, there's a picture right here of a nucleus that undergoes basically the same processes that we talked about in the previous lecture. So there's basically the cells, they shrink in size. This is the first thing. And in an H&E stain, hematoxylin and eosin stain, the cells, they actually become eosinophilic. And those followed by a fragmentation of the nuclei. So you can see right here that the nucleus is actually fragmented into two. Okay, and this is called karyorrhexis. And of course, this is karyolysis because once the chromatin condenses, it fragments, and then it is followed by a dissolution of that nucleus. Okay, so this is and it is actually difficult to actually see apoptosis within the histological slides. Now, what keeps cytochrome C within the mitochondrion? Of course, there is this gate right here, and every gate, there has to be a guardian of that gate. So the guardian of that gate is a molecule. Okay, it is a really important molecule. It is called the BCl2 molecule, and this acts as a guardian, and it keeps the cytochrome C within the mitochondria. Now, there are mainly three pathways by which apoptosis can occur. Now when cells get injured, basically when a cell gets damaged, the DNA is damaged by radiation. Oh, I'm sorry. When the DNA gets damaged by radiation, or by toxins, or by free radicals, and so on and so on and so on. Basically, when proteins are misfolded, or when there is a lack of a growth factor that is required to keep the cell viable. Now, what happens upon cellular injury, and why it triggers apoptosis, is that there are sensors that for the BCL2 family, and basically, there's another slide right here. In a viable cell, you can see right here, in a viable cell, that those sensors, they produce those anti-apoptotic proteins such as BCL2, and there is no leakage of the cytochrome C. However, when there is a cellular injury upon irradiation, upon misfolded proteins, or upon basically a lack of survival signals, a lack of hormones, basically what happens is that those sensors, they sense that cellular injury and they antagonize the action of BCL2. And then they will basically, the cytochrome C leaks out and the activation of caspases occurs and leads to apoptosis. So, the same here. Basically, the BCL family is antagonized, the mitochondria leaks the cytochrome C and the caspases, and we've talked about the breakdown of the cytoskeleton by proteases and by endonucleases, and the breakdown of the nuclei. Now, they will form something that is called a cytoplasmic bulbs, which is basically the cytosol and a few organelles, and once it buds off, it forms something that is called an apoptotic body. And the apoptotic body has a receptors or ligands that bind with phagocytes or with macrophages, and they are phagocytized. And this is why we don't get an acute inflammation after apoptosis. Now, the other pathway is basically a receptor-ligand interaction. So a molecule comes into the cell and tells the cell, go die, go kill yourselves. And I want to emphasize the TNF receptor, or the TNF family, the tumor necrosis factors, because the Fas ligand is actually a part of that tumor necrosis factor. So in tumor necrosis factors, they bind to a ligand, they activate some proteins that initiates the caspases the same way that cytochrome C does when it leaks out of the mitochondria, and the rest of apoptosis follows. And the reason that FAS is actually important is because, as an example, you should take the T cells that they are produced within the bone marrow, but they go and get educated within the thymus. Okay, and so when they get educated within within the thymus, they will undergo two questions, and we call them positive selection and negative selection. So in positive selection, they check if they can bind to self-antigens, and in negative selection, they check how avidly they can bind. So if cells, they bind too avidly to self-antigens, they are basically, they encounter the FAS and molecule, and they undergo apoptosis in that matter. So the FAS is important in the thymus education, because of course when they bind too avidly, we get autoimmune diseases. Okay,